Okay, so thanks everybody for coming to the Alliance Office Hours webinar. Today, we're very happy to have with us Dr. Tim Schedl, who is the Chief Science Officer for MicroPublication. And Tim is going to tell us a little bit about the journal and its scope and its purpose. So, Tim, I'm going to turn it over to you.
Okay, thank you, Carol. Let me share my screen and get things organized. Uh, okay, so micropublication biology is a novel publication platform. It's scientific community-based, nonprofit, and open access. All publications are PMC, European PMC, and Google Scholar indexed, discoverable in PubMed, and receive a citable DOI. So micropublication biology grew from a discussion that occurred at the end of a worm-based scientific advisory board meeting. And we had three main goals. The first is to increase the entry of peer-reviewed research findings into the public domain, employing a short article format. The second is to provide a peer-reviewed publication platform for research performed by undergraduates. And the third goal is to facilitate efficient incorporation of new findings and information into scientific databases like the Alliance. So what are the categories of findings for micropublication biology that are published? So of course, new findings, novel discoveries, new reagents, and/or methods. But in addition, we publish findings that other journals don't find, many other journals do not publish. And this includes negative results, replications that are successful, and this includes situations where one is scooped and beaten to the press by another laboratory, and replications unsuccessful. Studies can be observational, or they can be interventional and mechanistic. So what is a micropublication? It's a scholarly, peer-reviewed, concise report of research findings presented in a single figure with the goal of rapid dissemination of these experimental findings to the community. So our publications have the same standard, different features to them, but there are two main differences. The first is we just have a single figure and it can have multiple panels. And the second is that instead of introduction, results, and discussion, we have a single section called description, which subsumes and contains that content. And so what I'm going to do now is just to go through an example micropublication so you can get a sense of their content and organization. And so the title of this micropublication from Kurt Warnoff's lab is MOC6, and the human ortholog is MOC-S2A, is necessary for molybdenum cofactor synthesis in C. elegans. So we start off with the authors, their affiliation, and then an abstracts section here. And there for the abstract, we have a 100-word limit. Then we have the single figure for this paper, and there are multiple panels. The authors are showing a sequence alignment in panel A, and in panel B, what they're showing is the gene structure for the MOC6, and then these are the CRISPR editing strategy and the resulting deletion alleles that have been obtained. Then the authors have three data panels. The first are data that's shown graphically. And then in panel E is a different set of data that's shown in a table. Okay, and of course, our micropublication publications are rigorous. And so, for example, there are statistical comparisons that are made, demonstrating, scientifically sound conclusions. And so for the figure, it has a detailed figure legend, which really then details each of the features of the panels that are shown, and then help to guide the reader through what the data is. And so then the next section is the description section, and typically that's about 500 to 700 words. So in this article, the first paragraph and the first few sentences of the second paragraph really are introductory materials. And then the rest of the second paragraph, the third paragraph, and the fourth paragraph really explain what is the experiment, what were the findings, and then discussion of those findings. And then finally, the authors end with a conclusion paragraph from the work that they performed. And so then the next section is the method section. And here for the method section, the authors really are describing the experiment so that other scientists can replicate and extend their work. For the method section, we actually do not have a length limitation. Next, there's a reagents table here, and since this study is a genetic study, the genes and the alleles are shown that have been used in the experiments and their source. And finally, the article ends with acknowledgments, references, funding information, author contributions, reviewed by, history, and copyright. So micropublications are scholarly and meet the standards of the field. They should be well-written, scientifically sound, and rigorous. Micropublication peer review is made quick because the articles are short. We have a simple publication criteria. Is the work experimentally sound? Does the data support the conclusions? Is there sufficient information to allow replication? Is the text well written? Are the findings of potential use to the community? Perceived impact is not a criteria. For articles written by undergraduates with mentor guidance, the review criteria is exactly the same. So as I mentioned earlier, one of our goals was to facilitate incorporation of new findings into the scientific databases. So here I'm showing you, a standard current publishing scheme. Authors write the paper and they submit their manuscript. Then the scientific editors read and evaluate the article to see if passes muster for being sent out for peer review. The peer reviewer reads and writes a critiques, and the critique is then sent to the authors. And then the authors go and revise their article. And then if the manuscript is considered to be well written and scientifically sound, the article is published. And then at a later date, the curators go in and examine the article and extract information post-publication. There are issues with this current publication model. Excuse me. Articles can have nomenclature errors, and articles can have omissions in reporting. And the problem is these errors and reporting issues are found post-publication. And in almost all cases, those errors and omissions are not rectified. And so the literature, is contaminated with papers that have errors and omissions. And additionally, the current pathway, there's a delay in incorporation. So to rectify these issues, errors and omissions, what micropublication biology is doing, it is collaborating with database curators to validate and vet the reported data and reagents so that nomenclature and data reporting meet the community standards. So what happens is that after peer review, database curator collaborators evaluate the manuscript and then send requests to the authors to revise to provide missing data or correct errors. And then when the article is published, the errors are there are no errors and omissions. And so this then corrects this problem in the current publication scheme. So this curatorial peer review supports rigor and reproducibility by eliminating these errors, and it's not performed by any other journal. Okay, so what are the similarities and differences between micropublication biology and standard scientific publications? So one similarity is that articles in micropublication biology have the equivalent scientific rigor, clear and scholarly written text, and as full-length articles in other journals such as PLOS Biology, Genetics, Development, Evolution, Molecular Biology, the Cell, Current Biology, and others. But this is within the context of our publishing model, a single figure and concise text. So a standard publication typically is a line of investigation that can last 2 or more years. It generates multiple results shown in multiple figures and tables, and typically there are a number of supplemental figures and tables leading to multiple conclusions. Now, it's still important for researchers to publish these kind of standard articles for promotion and obtaining national grant funding. Now, a micropublication is a concise report of experimentally sound research findings presented in a single figure and essentially could be considered as a subsection of a standard publication. So when should you micropublish? And so I'm going to go through a number of examples. So if you have a successful pilot or circumscribed project, why not micropublish? If you have findings, reagents, or a method of value to the community now, but the project, which it is a part of, won't be published for a year or more, why not micropublish now and get this information out to the community? Often, there's lots of data that we have in our labs that might otherwise not ever be published. For example, you can have useful and concrete findings, but the project was discontinued or unrelated to current work or funding. Why not micropublish these findings? You might have a solid result that does not make it into the relevant publication or was included as data not shown. Why not micropublish? You may have a solid finding, but the student, postdoc, or technician has left the lab and it's not clear when the project would be followed up. Well, you may have findings that you can micropublish. Additionally, you may have there may be standalone findings that is part of a larger effort, but instead of being the 6th author for supplemental figure 10, you could be the 1st author of a micropublication. Now for undergraduate research, if you have done a project under PI mentored guidance, why not micropublish that? Also from an undergraduate course research project, you may be able to develop concrete findings. Why not micropublish those? Okay, so who are micropublication authors? They are mainstream research from major universities and colleges. So from the senior authors of micropublications, we are currently finding that 69% are from R1 institution and 31% are from non-R1 institutions. And among our authors are National Academy of Science members, HHMI investigators, and Nobel laureates. Okay, and so, we have seen continued growth in the number of articles published by Micropublication Biology. This is data from PubMed. And, since the work in C. elegans, has really grown out, since the work in micropublication biology really has grown out of C. elegans and worm base, it's not surprising that a large amount of our publications are from C. elegans. But importantly, as of November 2025, more than 1,500 articles have been published. And for C. elegans, as of 2024, 3% of all articles are actually micropublications. And so we're hopeful that other mods will have increased submissions from these for micropublication biology. And so here I'm just showing some example micropublications. So here are some Drosophila micropublications, mouse micropublications, budding yeast, zebrafish, and Xenopus. So now there were a few micropublications that occurred before the start of micropublication biology. And here's an example of a classic micropublication from Watson and Crick. So the success of micropublication biology, depends upon, an excellent team of editorial team. And so here I'm showing you our editorial team. This here we have C. elegans, Drosophila and Arabidopsis editorial staff. Here we have fish and yeast, zebrafish, dictyostelium, budding yeast, maize, structural biology, soybean, fungi, ecology and evolution, ciliate, and computational biology editors. And we have biochemistry, microbiology, and rat editors. And of course, we've got our curatorial staff, and this includes worm-base, soy-base, tetrahymena genome database, POM-base, mouse genome informatics, CottonGen, rat genome database, and zebrafish. And then here is the core editorial team. And I'd be happy to take any questions.
Thank you, Tim. One question, actually a couple of questions. One is in the reagents section that you showed, is there a requirement that those reagents are available for distribution, or is it just to describe the reagents?
Yeah, I think, in modern day science, I think the expectation is that that all biological reagents that were used in a study should be available to the community. For the model organisms, we have the advantage of there being stock centers. And so, for example, in that reagents table, it indicated which strains were available at the stock centers.
Okay. And then my other question, is there any thought about sort of opening this up to descriptions of software tools, for example?
Yeah, we actually do have some micropublications that are software tools. So so that's also part of micropublication biology.
Is there any special requirement for publishing a software product in the, like it has to be available, or?
Yeah, yes. So we don't publish articles about commercial software that you would have to pay for. All of them, are expected to be in the public domain. And, and certainly the any author who's considering, considering publishing software, they should feel free to, contact MicroPublications and just ask about it and what things could be done to facilitate the manuscript.
Great. I see Daniela has her hand raised.
Yes, thank you. Thank you, Tim. I just want to mention that we also publish database updates because this can be relevant to some of the curators that are here in the room. So we publish novel features or tools in existing databases or updates to databases such as new coverages of data. So consider to submit. And I don't know if you mentioned, Tim, but the article processing charges are very reasonable. So it's $350 per submission because we are a service to the community and nonprofit.
Thank you, Daniela. That was going to be one of my other questions. So you've already answered it. Great. Any other comments or questions from anybody else on the call? You can either raise your hand or just unmute yourself and ask your question.
Hi, this is Christina. I'm one of the on the editorial board as for the Xenopus section, and we're trying to encourage more Xenopus researchers to use micropubs. I just wanted to know if anyone on the call is a teaching faculty, and if they have or are they considering using micropubs, incorporating that into their teaching.
So I think that's a question for those on the call.
Correct.
There's Darren. Darren, would you like to say something?
Absolutely. Thank you. And thank you for giving this presentation. I work with undergrads and mainly teaching with a little research. I do some work with the Genomics Education Partnership, and we have been publishing gene models using the micropublications. But it's also something that I'm looking at now as, I've got undergraduates running projects. They get limited amount, if particularly if they join me in their senior year and are doing little, adding on to a project, they graduate before any of it sees its way into any kind of manuscript. So this seems to be a really good way to not only have them see their work translate into a publication, but get them writing about it in a way that's beneficial to their education. So I see it as a win-win. They actually get to do more of the writing about the work in advance. So yeah, thank you.
Thank you, Darren.
Yeah, Christine, I'll just say that I've alerted the education department here at the Jackson Laboratory about this, about micropublication, so that they can point our trainees to it as well, because I do think it would be, there are a lot of studies very much along the lines that Darren was just saying for mouse that I think are ideal for micropublication. So I'm hoping we can raise the awareness of this publishing opportunity for our trainees.
Excellent. I agree.
Anything else from anybody? All right. Well, thank you.
Yes, and thank you for your attention.
Yeah, thanks, Tim.
Thanks, Tim.
We will be posting this so that folks that couldn't make it today will have the opportunity to view this at a later date. So thanks again, everybody.
Thank you.
Bye-bye.